Hi class, so in this fourth video of chapter 2 we're going to talk about membranes. So the membrane is the outermost layer in a animal cell, and a plant cell is the second layer from the outside. So if this were an animal cell, this would be considered the membrane. The membrane provides protection for the cell, but also regulates what comes in and out of the cell. So the first assessment statement in chapter 2, section 4 is draw a label diagram to show the structure of membranes. So in order to draw a membrane, you need to know what a phospholipid is. So a phospholipid has two parts. It has the phosphate head group, and it has the lipid tail group. So this part is the lipid part, and this part is the phosphate part. The phosphate part is hydrophilic, and the lipid part is hydrophobic. So they're both characteristics in the same molecule. So in a cell membrane, you have lots of these molecules with the tails facing inward, and on the next assessment statement we'll talk about why it's faced this way. And inside this bilayer, this lipid membrane bilayer, there's two layers, are embedded are proteins, and there are proteins that span the membrane, so they're called transmembrane proteins. And there are many functions of transmembrane proteins, and we'll talk about those on a future slide as well. If we look at an artist representation of a cell membrane, you can see that there are lots of phospholipids. There's two layers, and you have proteins which go through the whole membrane, and those are called transmembrane proteins, or in this case they're called integral proteins, meaning they integrate themselves into the lipid bilayer. You have peripheral proteins, and peripheral proteins are only found on one side of the membrane, either on the inside on the cytoplasm or on the outside in the extracellular fluid. You also have some carbohydrates that are attached directly to proteins, and these are called glycoproteins. So these little hexagons are called carbohydrates, are the monosaccharides, and they're linked together, they're polysaccharides. The prefix glyco refers to carbohydrates. So a glycoprotein is a protein with carbohydrates attached. A glycolipid is a lipid here with carbohydrates attached. And in this picture you also see that there are cholesterol molecules that affect the fluidity of the membrane. So next assessment says, explain how the hydrophobic and hydrophilic properties of phospholipids help to maintain the structure of cell membranes. So if we draw our phospholipids, we realize that if there's water on the outside of the cell and there's water on the inside of the cell, you can't have just one layer. So what happens is that the tails will not want to be near the water because the tails are hydrophobic. So the way that you can create a friendly situation for the phospholipids is that you have the polar heads facing out towards the water outside the cell and the water inside the cell. So by having two layers, again with the tails facing inwards away from the water and with the heads facing outwards towards the water. In the next assessment statement, it says, list the functions of membrane proteins. So, one of these is a hormone binding site. So often, this is shown like this is the cell membrane. You have membrane protein sticking out, and there's a little cup that's waiting for a protein to sit, because this shape fits into the structure of the membrane protein. So imagine this little circle is a hormone. It binds to the hormone binding site, and then it transmits a signal into the cell to express a certain gene, for example. The proteins that are in the membranes can also be enzymes, and obviously we've talked about those. They catalyze chemical reactions. You can think of the membrane proteins as being involved in cell-cell adhesion. So for example, if you have another cell here and then one cell here, you can have two membrane proteins that are connecting here, and now it's causing these two cells to be attached. They can also be involved in cell-cell communication. Basically, one cell is telling another cell maybe that it's present, so you can stop growing. This is something that happens in your skin cells, for example, to make sure that your skin cells stop growing. The membrane proteins are also involved in passive transport and active transport. Passive transport is when things get moved from one side, from inside to outside or outside to inside without energy. And in active transport, you have to have energy. Energy is required. So, in the next statement, it refers to two methods, diffusion and osmosis, and both of these are passive transport because they do not require energy. So in this picture, I've shown that you have red particles in a container of water, and they will spread out from high concentration over here on the left to low concentration over here on the right. So this is called diffusion. So diffusion is the general movement of particles from high to low concentration. When you're referring to water, the movement of water from high concentration to low concentration, that's called osmosis. In this picture, it explains passive transport across membranes by simple diffusion and facilitated diffusion. Just ignore this part over here for now. So simple diffusion is, you can see that the particles move from high concentration to low concentration, and it doesn't require any proteins. However, if you're using a transmembrane protein or an integral protein, and it flows from high concentration to low concentration, that's called facilitated diffusion. So facilitated diffusion uses proteins. So before we go on to the next section, you have to know what ATP is, because the book never really discusses it, and so I want to give you a quick little preview. ATP is a form of chemical energy. Basically, when your body is ready to use the energy that it has inside of its cells, it converts it to this form of chemical energy because it's very short term. It holds it in its bonds, so it can be used later. So ATP has three parts. It has a ribose sugar, it has an adenine base, and it has three phosphates. That's why it's called adenine triphosphate. Three phosphates. And in this diagram over here on the right, you can see the three phosphates are drawn with three little p's. When you break this last bond right there, you release energy. And that energy can be used to drive chemical reactions. So you can see here, when you break this bond right there, you see that you have two phosphates left. Now this is called ADP. And you can also break that bond and make it AMP, adenosine monophosphate, and you can release more energy. You can put energy in the reverse process and get ATP back. Okay, so now ATP is used to drive active transport, which is this part over here. So notice that if the gradient, meaning the kind of the slope of particles going from high to low concentration, is going from the top of the picture to the bottom of the picture here, well, to get the molecules going the other way, you have to add energy. It's kind of like thinking about it as pushing the ball up the hill. If you want, the ball will roll down the hill by itself, but to make it go up the hill, you have to put energy in. And that's what the ATP is doing right here. It's pushing the concentration against the gradient. So an example of this is the sodium-potassium pump. Now later on in the class we'll talk about the importance of the sodium-potassium pump, but what you have to know for now is that potassium has a high concentration inside the cell, and sodium ions have a very low concentration inside the cell. Now if we look at outside the cell, the sodium is a very high concentration and potassium is very low. And what the sodium-potassium pump does is it takes the sodium that's very high in concentration outside the cell and putting more of them outside. And if there's a lot of potassium inside, we're taking the potassium that's outside the cell and putting it inside. So we're pushing it against the gradient. And that's what this says, these words say here on the side. You should pause that and take a chance to read these notes. Okay, so how does this work? Well, if we take a look at this picture on the top left, the sodium ions bind to the inside of the transmembrane protein. ATP is sitting here. When ATP binds to the protein, it releases energy when it becomes ADP, and there's a conformational change that occurs in the protein. And this conformational change causes the release of the sodium ions into the extracellular space outside the cell. Then, when those sodium ions are released, the potassium ions can bind to these empty slots inside the transmembrane protein. When it does that, the phosphate gets released and the transmembrane protein goes back to its original conformation, allowing the potassium ions to be released into the cell, so that this process can happen again. Again, we're getting the sodium ions out of the cell towards where their concentration is already very high. And we're trying to get the potassium ions inside the cell where the concentration of potassium ions is very high. Basically, this drives a lot of other chemical processes that occur inside your cells that we'll talk about later. In the next assessment statement, it talks about how vesicles are used to transport materials within a cell between the rough ER, Golgi, and the plasma membrane. So, here is the endoplasmic reticulum with the ribosomes on it. It's called the rough ER. Here's the plasma membrane, and in between is the Golgi apparatus. So you can see that when you make a protein on the ribosome, it travels through and gets folded properly in the ER, and then it bubbles off. Part of the membrane bubbles here, and it transports it down towards the Golgi apparatus. It fuses with the cis Golgi, the cis face meaning it's closer to the nucleus, and each little stack in between bubbles through and rebinds to the next layer of the Golgi. It fuses with it and then releases another vesicle on the trans face of the Golgi. Then it travels towards the plasma membrane and fuses again with the plasma membrane and releases its contents to the extracellular space. I strongly encourage that you read these notes on the side. So in the last assessment statement of this section in chapter 2, it says, describe how the fluidity of the membrane allows it to change shape, break, and reform during endocytosis and exocytosis. So, the word endocytosis refers to molecules being absorbed into the cell. Exocytosis is when you're releasing particles outside the cell. So, in order for this to happen, the membrane has to be fluid. It has to be able to change shape and to be able to move. So you can see in this top picture here, this is endocytosis. These molecules are being absorbed through an indentation in the plasma membrane. It pinches off more, and then it fuses it so the membrane becomes a little bit smaller as a result of this vesicle being made, and then this vesicle will be transported somewhere in the cell, so maybe the parts can be broken down maybe by the lysosome. In this picture, this process is called exocytosis. So the molecules are being moved towards the surface, the vesicle fuses with the plasma membrane, and when it does that, the plasma membrane becomes slightly larger because it's being added to, and then those particles that were inside the vesicle get released into the extracellular space. So I hope this was helpful in helping you understand section 2.4 on membranes. Thanks. Bye.